Thank you for coming, and if you'd like to move forward, we're not showing any slides, so you don't need to see the slides. As you know, plant, animal, microbial engineering using transgenic and genome editing offers an opportunity to incorporate many economic and agronomic benefits and that are difficult or impossible to achieve using traditional breeding. And recent advances in genome editing has really caused an explosion of interest and activity. The regulatory climate around genome editing, however, is very confusing at the moment and is still developing. And it affects how these products may be traded, etc. It also affects the research that you're doing. This panel will discuss how the regulatory environment is affecting and will influence future developments. First, I'd like to appreciate support from the USDA Biotechnology Risk Assessment Program, or BRAG, that supports generation of information that will assist science-based decisions on genetically engineered organisms in the USA. We have 4 excellent panelists, so I'll get them to introduce themselves in a minute. I also wanted to introduce Adi Alt-Holland, my co-convener over here in the corner. She's a faculty member and cancer researcher at Tufts University in Boston, Massachusetts. And she's representing the In Vitro Animal Cell Sciences section of SIVB. We will have introductions from the panelists, and then I'll ask them a few questions to get things going. I do expect questions from the audience. That's what we're here for. Please note this session is being recorded for later viewing by SIVB and other members. So please make sure that you go to the microphone in the middle of the room to ask questions, first stating your name and affiliation. If you need help, Dr. Sarbash from Mandabhan Bandari University in Nepal is here to help you. He's our student. If you'd like to stand up, just raise your hand. Yes, to help you. To start, myself, I've been around this system, doing tissue culture and plant genetic engineering for what, 45 years? Started in tissue culture, so that's my home. In Leicester, England, I don't know if anybody here knows of Professor Street, who was one of the pioneers in that area. Anyway, after a stint in Leiden in the Netherlands, doing agrobacterium, and then with Dr. Potrikas in Basel, I came over here to work on maize protoplasts, and which we published in '86, and then was hired to build a regulatory group at a company called Agrivo that has been absorbed into God knows how many companies. Recently, I really focused on method development and for tracing these things in trade, and also then on genome-edited products. So I'd like to introduce the panelists, and in order, I guess, Alison is to my left. You start off with a quick introduction of yourself, please.
All right, good afternoon, everyone. My name's Alison Van Eenenum, and I am a professor at the University of California in Davis, and my speciality is animal biotechnology and genomics, and I feel a little bit out of place at an in vitro meeting, because actually we do things in vitro, but actually I work mostly with live, genetically engineered or genome-edited animals, and I've been at the university about 25 years, and before that, to go to an origin story, I actually started off at Celgene as a student intern when I was a PhD student, the year they released the Flavor Saver tomato. And so that was a really exciting time. That was before everybody hated GMOs, so that's cool.
We don't hate GMOs. By the way, I just got a reminder from the hotel to turn my phone off, so maybe everybody in the audience can as well, because it dinged. So, okay. Wayne.
Good afternoon, everyone. Wayne Parrott, professor in crop and soil sciences at the University of Georgia. My lab focuses on development of tissue culture protocols and their use for genetic engineering and for gene editing. I've been at Georgia since '88, so before a lot of you were born. And I got my start as an undergraduate in a tissue culture lab at Kentucky with Glenn Collins.
There we go. Okay, so I'm John Kortz. Currently I'm working as a consultant, but previous iterations in my career, I've worked in a lab doing tissue culture. I think many of us started off doing tissue culture. I worked in regulatory probably for the last for 10 years toward the end of my hard-working days. Anyway, I was a regulator in USDA APHIS, Biotechnology Regulatory Services. So I dealt with petitions for deregulation. I dealt with permits. I dealt with notifications. Notifications are back in play now. And currently I do consulting for people who need or want assistance getting through the regulations, either primarily at USDA or FDA. So you may see my name on a couple of documents that come out from those agencies from now periodically.
Thank you.
Hi, everyone. I'm Nicole Juba. I'm currently the Senior Director of Regulatory Policy at the Biotechnology Innovation Organization, and I represent a portfolio of member companies that work in biotech plants, animals, and microorganisms. I've been living what I call the biotech regulatory dream for about 20 years. I came into it through peanut tissue culture and transformation as well. And in the course of the last 20 years, I've probably submitted dozens of petitions, registrations, permits for a whole host of crops, microbial products in probably about 20 different countries. So really appreciate the opportunity to speak with you all today.
Our first question goes to the heart of something that happened at the end of last year. And what was the court decision of December 2024 on the USDA secure rule regulations, and what are the effects of the return to previous regulations on research? And who would like to answer that? John, maybe? Or,
sure.
Okay.
As people are probably familiar, there is a biotech regulatory system within the U.S. The question's focusing on USDA, and prior to this past December, we were operating under a revision to the regulation for biotech or genetically engineered organisms at USDA that we called the Secure Rule. The Secure Rule created certain opportunities for streamlining products of biotech that could have been produced equivalent to traditional breeding, but also created additional pathways for how you can commercialize products that were otherwise created. There was a challenge to that regulatory process right after it was released. And just this past December, the court in California decided to vacate the Secure Rule and go back to the regulations that were in place prior to May 2020. This created different opportunities and challenges for research, for commercializing products, and really took us down a path of going back, but trying to also at the same time keep the learnings we've had over the last 5 years, particularly for genome-edited products. What this means is we live in a world where you can now, once again, ask USDA, am I regulated, and see if the products you're working on in your lab have to have some regulatory oversight for the research activities you're doing. And if USDA says, yes, you do, there's now various processes, like John mentioned, notifications, permits, and other pathways that you might then have to follow. Anyone want to add?
No comment? Yeah.
So let me just add a couple of things. The previous iteration of the regulations, I was at APHIS when the previous iteration was around. So there were notifications, permits, and then petitions for deregulation. The biggest change that I see is that under the Secure Rule, you could ask for what's called a confirmation request. And most genome edited products would fall under that. You could put in a relatively abbreviated submission to the agency, and the intent was that they would be able to approve that within 3 to 6 or 9 months. Prior to that, prior to the Secure Rule, the process was these petitions for deregulation, and those toward the end of 2018, 2019, when those regulations were finally getting phased out, that was taking anywhere from 6 months to 2 years or more to get through that process. So now that process is back in place, all right? So that petition for deregulation. For those of you who are signed up for the stakeholder newsletters, you saw a few things come out this week for an orchid, a corn product, and the blight-resistant chestnut. So the petition for deregulation, it takes a number of steps within the agency, and most of those steps are fairly public and invariably bring a lot of comments from the public and from those opposed to genetic engineering. So we're back to that now instead of a confirmation request and what would otherwise be a relatively quick approval process from USDA. The good thing, one of the relatively small good things is that under the secure rule, everything was going under permit. So prior to that, things for field trials and movement of things could fall under the notification system. Notification system is invariably a little bit easier for managing permits and things for moving organisms and for field release. So that's kind of a good thing under the old rules. So we can use that anyway. But there's a lot of details that we can talk about.
Just speaking as a user, someone that has to apply for this. The original rule, the one that's back in place, was very heavy-handed on transformation, but not so much on editing. When the secure rule came out, it really aligned what you had to do for transformation with what the known, any known hazard, known risk was. So it really lightened up the workload to get a transgenic either permitted or approved, but it was way too heavy-handed on editing. So we're back to if you're edited and you don't have any foreign DNA in there, you can self-determine. You don't even need to ask USDA if you can transport this or plant it outside. So from that perspective, things have improved for editing. They have not improved for transformation.
Any other comment? Of course, that may affect being able to understand where field trials and stuff are, but we'll come to that later. In light of that, can you share what the biggest regulatory challenge is for those working with edited organisms briefly? Wayne.
We've been focusing on USDA regulations, but EPA regulations, if you use editing to obtain disease resistance, you're going to be, you have to get an exemption from the EPA to treat you otherwise they'll treat you like a pesticide. And above and beyond regulations, we can't forget the sensibilities of the societies around which we operate. And we still think the day there's going to be sooner or later an edited seed is going to show up where it's not wanted and it's going to cause a lot of problems. So I guess that'll fall more under the stewardship discussions we're going to have.
So Alison, for animals, how does that go?
Do you want to do plants?
What's the challenge?
I'll do animals. So animals are quite differently regulated to plants, and this has been true whether it's genetically engineered organisms or genome edited organisms that can have a single SNP change. If it's intentionally done by a human, then it's regulated as a new animal drug. And the FDA basically says that's an unapproved drug and that can no longer enter the food supply without an approval process. So in order to get that, if you're going for full new animal drug approval, that's a multi-generational, multi-million-dollar process. There is something called enforcement discretion that some products are eligible for if you've mimicked a mutation that exists in the natural germplasm of your breed, of your species, so maybe it's in one breed of cows and you've moved it into a different breed of cow, then you can apply for this thing called enforcement discretion, which is kind of a wonky term for a low-risk product that the FDA is choosing not to exercise regulatory enforcement on, which is kind of like the CHP officer telling me that he's not going to give me a ticket because I was only going 57 in a 55 zone, which is true, but they could change their mind next week or if a new administration comes in. So that doesn't give me a great level of security. And I guess for me, my brain will not allow me to accept that genetic variation is a drug. It just, like, no other country thinks that.
And then I was just going to address on genome-edited microbes, which I think we're seeing more and more come through research pipelines, commercialization pipelines. Yes, with the vacatur of the secure rule, going back to the legacy Part 340 process, there is a much clearer path to commercialize a genome-edited microbe that does not require petition or permitting through the biotech part of USDA. But then there's also the plant protection and quarantine part of USDA, who has different oversight and different requirements. There's EPA, depending on if your product has pesticidal qualities, you'd fall under FIFRA. If you're a new chemical, you fall under TSCA. So even if you've figured out how to navigate with your genome-edited organism through the biotech regulations in the United States, there's still other agencies that could also have an interest in your product before you can freely use it how you want to without that oversight. So just something to think about.
Okay, thanks.
Let me add one thing. For those practitioners out there who are doing genome editing, under the secure rule, the rule that was vacated, there was a process, confirmation request, not too complicated. Send in some basic data to USDA, APHIS, BRS, and they would be able to approve something fairly quickly. Under the old rule, which is what we're dealing with right now, the process it's called an am I regulated process. So this is very useful to those doing genome editing. They're using CRISPR-Cas9 where there's no leftover gene sequence or anything. Fundamentally, the am I regulated process is very similar to what you would submit to the agency under the confirmation request process. So for those looking to commercialize or get out from under regulation, you're doing genome editing where there's no leftover gene sequences, no sequence from agrobacterium or other plant pests, you can still use this am I regulated process and it's not overly cumbersome. And they do have guidance on their website about that.
Thank you. I think one of the things I'd like you to clarify is at what point, and that goes for all of these organisms, in your research, do you have to go to the agency? Because we talk about this and then people are going, well, if I'm going to make an edit in this tube, do I have to go to the USDA or FDA? So can you clarify for us, and I see Alison's ready to go, can you clarify for us at what stage do I have to go to the regulatory agency, at least in the US, we're not talking global because that's a whole different ballgame, but what stage do we actually have to do something in tainted paperwork?
So if you make an edited animal that's alive, running around, then that technically falls under FDA. They will give an exemption to all rodents and biomedical type animals, but even me at UC Davis, theoretically, if with my edited animals, I would have to do a petition to the FDA. I've done the math on trying to get what's called food use authorization for my animals, where I've got a knockout that does whatever I want it to do, and then normally those animals would go in the food supply and we sell them in our meat lab at the university. Because they're an unapproved drug, I have to either get a food use authorization or an approval for them to enter commerce. If I don't get either of those, then I would be technically dealing in illegal drugs. I'd become a drug trafficker and I could get prosecuted for drug trafficking. So I just incinerate all my animals.
Yeah, unfortunate.
So for USDA and plants, so Wayne mentioned this voluntary self-determination. If you have genome edited products that don't have any plant pest sequences in them, there is this self-determination you can do, but you better have good documentation about it. You better have something in your file that documents that what you've got doesn't contain any plant pest sequences. If you're uncertain or don't want to take the time to do it, when you want to move a product across state lines or release it into a field trial, you need approval from USDA under either a notification or a permit to do that. And it takes a little bit of time to get those. It's a process.
I was just going to add, if I was in your shoes again, like I was 20 years ago, the first question I would ask myself is what I just generated in this tube or in this dish or in the greenhouse, am I going to move it across state lines? Am I looking to bring something into the country to import it? Or am I looking to release it into the environment, even if that's the environment in the same state my research institution is in? And if the answer to those are no, I'm not looking to move it, I'm not going to import it, I'm not going to release it, then there's no oversight, at least from USDA's perspective. And then you can start asking yourselves those other questions and putting that documentation together to say, do I think I'm regulated by the USDA? Do I want to ask them? Do I want to self-determine? But those would be where I would start: import, movement across state lines, or release.
Yeah, so if there's most of the stuff I do is either going to be a notification or a permit. So if we have any intention of even doing a field trial within the state, we would certainly apply for the paperwork 6 months before we're ready to start the trials. But if your goal is to commercialize a product, the marketplace is going to have requirements above and beyond the regulations as well. So they're not going to settle for a self-determination in most cases. So again, what you ultimately intend to do with the product does come into play, and you'd probably want to consult with FDA and get guidance from them, as well as if it's a resistance trait or pest resistance trait, EPA, and of course USDA for everything else.
The next question I have is for the audience. How many of you have submitted something under one of these rules?
No, I have. I wanted to have a question because
Yes?
self-determination, and so generally these applications for APHIS, it is assigned to an individual person, right? PI or things like that. And then I wonder if there is a system, which I am not aware, that's why I'm asking, is there a proxy opportunity for a person who is supposed to self-determine? Just like, tax, I'm filing a tax, but I can go to somebody who can file on my behalf. So is there something like that system exist, you guys know?
So I have filed, as a consultant, I have filed things on behalf of somebody else. So I filed I guess it was a confirmation request originally, but it turned into an am-I-regulated submission to USDA for a company in Israel, and I didn't get approved under the confirmation request under the new secure rule in a timely fashion. Then the lawsuit happened, and the regulations were vacated. So it ended up getting approved under the Am I Regulated letter a month or two later. So I'm, as a consultant, now the person who needs to submit, I believe you have to be a U.S. citizen in order to submit these sort of documents. So I can do that on behalf of other people, and approvals can take place. It's not complicated. In terms of getting permits and things through the agency to do field trials or move things or import things. Generally, the agencies don't like postdocs. They like permanent employees or people who are known known entities that are going to be in a position for some period of time to submit.
Yeah, I mean, in companies you have reams of people to do this. A lot of people in small companies or in universities don't have the backing, maybe even of your center. I said the other thing I wanted to do was is there a question? Okay, thank you.
Yeah. First of all, I want to
Could
thank
you
for,
say your name?
So Alan Van Dynes at UC Davis.
Thank you.
First of all, I want to thank you all for being on the front line. I've done a lot of work in this space too, and I've spent too much of my life on it, as well as we all have. And I want to commend Allison. Everything we do in plants is just buried compared to anything you do. We can incinerate plants, but it's not that hurtful. My question is with the, with the new old rule, the secure rule was clearly going down the wrong way for gene editing, limiting us to one gene or homologs from that one gene, and that's bad news. USDA has appealed that decision, and does anybody know where that is right now? And then kind of where are any of you, or maybe Nicole especially, what are you all doing to try to put them back on the right track? Because the rest of the world is moving on, really taking on kind of the Argentinian rule. Most of South America has, at least 12 countries in Africa have, 26 are about to do it. So, yeah, are we on track on this, or are we just going to go down the deep end again?
Yeah, I think we're back on track. It could be better. What Alan's talking about is when the court ruled to vacate the secure rule, the USDA issued a notice that they were going to appeal that decision. And Bio and another association were interveners in the case, so we were actively involved trying to support USDA. And at the 11th and a half hour, we were able to convince USDA to withdraw the appeal request because, as everyone sort of acknowledged, there were some more efficient processes through the legacy regulation for products of genome editing. So I think the risk of an appeal and a overturn of the vacatur to go back to the secure rule is not going to happen. There's still a few things administratively USDA and the plaintiffs have to settle out in court. But it still could be more efficient for products of traditional transgenesis that we wanted to see also gain efficiency. And I think there's opportunity for regulatory reform there that USDA is actively considering. How they will do that, I think we will just have to see.
Thank you. Any other questions? Okay, so the next question that actually the panelists wanted to ask is, audience, raise your hands if you use any stewardship practices, that is containment of materials, in your laboratory or the field. And I hope I see a lot of hands. Is there anybody who's doing this and not using stewardship and needs some advice? Probably. Okay, so focusing on this containment, what stewardship initiatives are in place for academics and for companies that have little or no experience? And I worked in a very large company that had a lot of experience and was actually involved in cleaning up the mess a couple of times from some incidents and taught incident management for 15 years. And also had to deal with some situations where a company that's coming into the field new may not be as observant of the need for certain stewardship practices. So what is being done to educate, not only to help you guys know what you have to do, but also small companies coming into this field, both in animal and in plant?
So I can start from the plant perspective. We know it can be really difficult to put together these really large programs, but we also understand how important it is for containment, whether you're at a really small scale research plot at your extension station or you're on 100 acres in the Midwest. We know that when you have requirements from USDA, they will dictate how that containment is being managed. But as we have products that are no longer subject to regulation and no longer subject to that oversight by USDA, it's incumbent upon all of us to put together good stewardship principles. At Bio, we work with an organization called the Global Stewardship Group, and they do put out these best practices. They're not requirements, but best practices for how you can contain material when it's out in the field, how you prevent it from getting into trade and other places it doesn't belong. And I do believe if you go to their website, they offer some free courses for academics that you can go and take and see those best practices and guidance. So that's where we would suggest people start.
Okay.
I've worked with the Global Stewardship Council trying to put some of those courses together. I know a couple of people in the audience have too. It was an exercise in frustration. I don't think they are where they need to be for academic labs. And if I ever have enough time, I can certainly put something together, or even if people want to help. But it is, I think the vast majority of people are really underestimating how fragile social acceptance is. And again, one seed where it shouldn't be can really set us back, set the whole industry back tremendously. So it is of crucial importance to get it right.
What about, is there
So there's not really anything quite analogous like that. Obviously, we have to contain our animals and double fence them and have 2 ear tags. That's the rules. So we do that. But I guess to me, the bigger concern would be for some of these, well, there's only actually 2 animals that have ever got it, but this enforcement discretion decision technically allows the meat, milk, eggs if it was a chicken, and gametes, as in semen and ova, to enter commerce because they're not going to exercise enforcement discretion. And to me, that sets up a bit of a risky scenario where can we guarantee there's no genome-edited animals in our meat supply? No. Now, I'm not saying those 2 are, but they technically could be. And so how does that happen? So the animal biotech gene editing is such a small community. It's really like 3 or 4 labs that are left, just the crazy people that have put up with this for 20 years. Whereas the companies that are doing it professionally, like Genus and things, they're going to have very, very good containment because they're a large multinational company. But there are other ones that are working in things like fish that potentially could get escaped, where I think they have, they're more of the species that I would be worried about than a cow. I'm not worried my cows are going to break loose and breed the world. But a fish could. But most of the fish are being edited to be infertile, so that's kind of ironic. So there was actually one lady in Europe that was trying to get permission to do an infertile fish trial because this was a way to contain genome-edited animals by having infertile fish, and she couldn't get the permit because there was a risk that they might not be infertile. And so, i.e. the normal fish that they're raising in aquaculture in Norway. So it was like, well, if that's a risk, then why are you allowing aquaculture? But yeah, there wasn't a lot of sense to that. But there's just not the same kind of large seed company stewardship kind of things that we have here. There's very tight stewardship around semen control. I'm talking bull semen here, and gamete transfer in terms of biosecurity and disease testing and all of that, but that's true for all semen. It's not unique to genome editing.
Okay. Yeah, I mean, we say there's stewardship in place, but there has been, as I know to my, that's how I got into this area of incident management. These things, you only need 1 seed to be in the wrong place, or 1 pollen. And that's one of the things you really have to be careful about once you start getting into a mixed greenhouse situation or into a field. And I think I talked about that 2 years ago, this meeting. Do we have some more questions from the audience, please? Yes, please.
Hello? Yeah. I know that we are in US, that you guys started talking about regulations here in US with USDA, FDAs, and my question here is more like, how do you guys are in collaborating or working towards other countries, like on what you have learned and how we can support them? Because I think that, we are pioneers, but like how is like, your relation with them and moving things forward?
Yeah, so when I was with USDA, there were efforts to work with other countries, mostly through the OECD. Now, all the countries around the world doing these sort of things are not OECD members, so sometimes they're helpful, sometimes they're not. And I honestly, I don't know. I've been away from USDA now for a few years, and I'm not sure. When I was there, we would literally go meet with regulatory officials in other countries. So we would meet with the Taiwanese, we would meet with the Chinese, meet with the Japanese. I think we didn't view most countries in South America as needing any help. They seem to be pretty able to move forward on their own. Now in the current administration, I'm not sure how that's kind of playing out. One of the things that I found, and I think at the time when I was with USDA, and I think it's still the case, in some of these other countries, China, Vietnam, I'm not sure about some of the other Asian countries, but the regulatory officials there oftentimes are appointed for like a 1 or 2 year basis, and then they leave. So as a full-time, the regulators in the U.S., they are full-time regulators, and they have job permanently doing this sort of work. So you invariably have to, you meet with other regulators, you educate them, and about the time, unfortunately, that they're getting up to speed, they move back to another job, and you've got new people to try to educate about at least about how the U.S. does it. So it's a little bit problematic when other countries don't have people dedicated to this sort of work. I'm not sure what else I can say about that right now. I hope that's helpful.
Just on that part too, so for transgenics, we used to look at the U.S. as having a really efficient system, and for transgenics, we would want other countries to try to base off of the science risk proportionate system we had here in the U.S. For genome editing, I think that we've seen a shift. There are countries like in South America, Colombia, Honduras, Guatemala, Argentina, Brazil, Costa Rica, Dominican Republic, who've been able to find a way to streamline their genome edited policies for plants, for microbes, for animals, in a way that the U.S. just has not quite caught up to, even with the vacatur and our reversion back to the legacy regulation, because as others have mentioned, you still have EPA, you still have FDA to deal with. We're also seeing countries that never had regulatory policy for GMOs skip that part of their regulatory process and now have a policy to handle genome editing. So I do think that's really interesting. And to John's point, these countries get together within government forums and talk about how they're doing this. But I do think for genome editing, we're seeing a lot of good progress outside the US. But maybe some of the larger geographies like Europe, China still have a little bit of a way to go.
On the animal front, the USDA FAS, Foreign Ag Service, actually sponsors a international workshop of regulations of animal biotech. We've got our 6th one coming up at ISBR in Ghent in November. And that brings in as many regulators as we can get together to try to get some sort of harmonization, for want of a better word, where at least we can enable trade, because if we can't trade meat and milk and eggs, then it's going to stop it in all countries. And that basically is an effort that we've had ongoing for almost over a decade now to try to get some harmonization there.
There was a question over there. Can we do that one first? He was up there a long time ago.
Hi there, I'm Freddie from Purdue University, doctoral candidate. I think keeping on with the theme of the previous question about non-US regulation, I'm curious the panelists' opinions about Europe and maybe more specifically the 2025 earlier this year kind of move by Poland to deregulate that EU space. I think the EU is often a trendsetter for regulation, and I'm curious what our panelists think about EU's future.
We certainly see that the European Union is developing a process to treat genome-edited organisms differently from traditional GMOs, and we do applaud that process. I think what that looks like in practice is going to be what we have to see. EFSA had recently put out, even for risk assessment of biotech animals, how they would go through and develop those scientific opinions. So we're really excited to see Europe thinking through that process. I think it's going to be a little bit of ways before we see what that looks like when it's actually implemented, and that's really where we'll be able to tell how efficient it's going to be. A small watch out I have just from some experience recently is we were seeing a lot of progress being made in the UK, particularly with their exit from the European Union. And now as there's more bilateral talks between those two geographies, we're a little bit hesitant to see if Europe's going to continue to have a bigger influence on the UK, and then maybe by extension some countries in other parts of the world. So I think that's just what we'll have to wait and see, but others can comment.
Just, the European Union is very complicated in terms of their process. So, they have to agree, not just the European Council has to agree, the European Parliament has to agree. So it's got many different moving parts. I'd say it might be more dysfunctional than our Congress, but even if it's as dysfunctional as our Congress, you can see what the speed of getting things through them is.
I think one of the things that about the European regulations and generally regulations is there's a lot of talk about a single base pair change. There's not a lot of talk about small inserts and deletions. And we're in danger of celebrating advance on the single base pair, and the other stuff gets stuck forever. And so I don't know if the panelists have got any suggestions or comments on that, or briefly to figure out, just address that issue, because we don't want to get that lost. It's a very important issue, and then if we're not careful, we'll get stuck.
Yeah, and I think it's not just going to be in Europe. I mean, that's where we see a big issue now. We saw the direction USDA was headed under the secure rule, where we were counting edits in diploid species versus polyploid species.
Yeah.
Experienced recently some feedback from the FDA Human Food Program here in the United States that their definition of genome editing maybe did not align with the definition industry was thinking meant genome editing. So as we're using processes and definitions of processes to determine our regulation, is that really getting us where we need to be versus focusing on the characteristics of the product and risk above the baseline of what we already have out today? So that would just be my comment there, Ray.
Yeah, and my comment would be that we really must focus on what the product is and not the process that we use to make it. So right now, if I make a lot of genome changes with X-rays, that's fine, but if I make the same ones with editing, it's not fine. That has no scientific validity at all. And our best baseline we have is comparing it to natural variation out there. And with the advances that we have had in sequencing, long-range sequencing and genome assembly, we're just now really getting a true picture of just how much standing variation is among our crops. I've seen papers where they've compared two soybean varieties, a Chinese one with an American one, and then they compared an Asian barley with a US barley with a European barley. In every case, when we look at, compare two varieties, we're looking at about 1 million base pair changes and about 1 million indel changes. So if you already have 1 million indel changes, what makes you think that number 1,000,001 is going to cause a problem that the first million did not?
The question I have...
Name?
Yeah, my name is Veena. I'm from Donald Danforth Plant Science Center. And what we have talked and heard so far is like higher level view, like these policies in place. Many times they are logical and sometimes they are not. And but they are there, and we have to deal with it. And many of the community members who are attending here, they are first time or beginners. And so why does these policies matter? And then why, people in the lab, like, oh, I'm changing one base. Why I should care about stewardship and compliance? Can you give the example, if we don't do that and things get out in the field or things like that, how it impacts and why countries care, why policymakers care. And so can you all speak, like animal side, plant side? I would like to hear your comment.
So for those of you who haven't been around for 35 or 40 years, and there are really only a handful of incidents that one can point to, but it's, maybe it's less than 10 large ones. But a couple of those were $1 billion incidents. And the mistakes that were made were what we would consider in this room fairly small. Things were mislabeled, things were mishandled. More often than not, there was human error involved. And that's why we talk a lot about processes, how you manage things and stewardship, and having standard operating procedures. Invariably, even if you have standard operating procedures or label things or this, that, and the other things can get mixed up, things can get misplaced, things can get mishandled. There's a Liberty Link rice incident. You can talk about $1 billion. Companies go out of business. Protogene was a company doing pharmaceutical production type things in corn, and they went out of business. They got a huge fine from USDA. Nothing was ever actually found in commerce related to the Protogene incident. But then there's BT10, BT11 incidents where things were made in a laboratory and they were field trialed, and they made relatively small amounts of seed, but the seed ended up getting mishandled. USDA got involved, FDA got involved, EPA got involved. Again, large fines, things that you get, particularly in a commodity crop. You can get seed infiltrating commodity crops at small amounts, and Ray can tell you how much a small amount is. That can be very difficult to get out of the commercial seed supply. So and it can take literally years to get those sort of small inadvertent amounts of contaminating seed out of the seed supply. Even if they don't pose a safety risk or a health risk, it's a problem.
So many, yeah, I would like to hear from animal side, but the thing is, whatever you mentioned, it is an impact. Many times people who are in research and academic workflows or academic researchers, so many times they are not making a product which goes to the market. However, we do follow these policies, or we are asked to follow. So many times people will say, it doesn't apply to me. And what I'm doing today, it's R&D, doesn't apply to me, but stewardship matter at that level. So what advice you would give to them that they should care about it?
We used to say, just as somebody and he mentioned the rice incident, the answer was 0.006%. And that's what stopped trade to Europe and various places. It's not much. And it was very expensive, and it took a lot of our time. I was on the cleanup, by the way.
I can give you an example of what happened to a professor where some null segregants, so animals that were not transgenic, that were housed with some pigs, were put into the food supply. So there was no genetically engineered pig went into the food supply, but at the time the FDA had the opinion that, I don't know, there was going to be horizontal gene transfer from one pig to another. I'm not really sure how that works, but anyway. And this professor it was like an ice raid. This is Matt Wheeler at Illinois, if you're curious who it was, and you can ask him about it. But he had like cops coming in and shutting his office down, taking all of his equipment. It was insane. It was just as if he had broken the he's trading unapproved animal drugs, and so it actually could come and bite you as a researcher, even though you think, oh, that's nothing to do with me. So that made everybody sit up and take notice that's in the academic community to make sure that we were not running afoul of what the law is.
Yeah, we used to say in the seed business, I'll come to you. In the seed business, we used to say, no seed left behind. Even if you don't know it's transgenic or not, you pick it up. Right? It's never left on the floor. Question. We got a question over there first, then we'll take you.
Hi, this is Daniel, a professor at the University of Maryland. I just want to follow up on the comment that you said about the types of edits. And so, as a researcher, I work with more complex editing approaches where we're targeting sometimes one region but multiple guides to create complex structural variants. And we're modifying regulatory regions to induce quantitative changes. But I'm always a little curious about, the types of approaches that we can use for generating these things, whether they will be treated differently as opposed to having one guide creating a single base pair or a smaller, less complex variation. So under the new rule or the previous rule that now is whatever, right? So can you elaborate a little bit on that? Because, I just want to know how we should approach this as we're generating these types of modifications that we would like to test in the field and, knocking on wood sometime, maybe even commercializing them.
Give it a shot.
It's complicated, right? Thinking, and you're doing the right thing. You're saying, hey, this is what I'm working on. These are the modifications I'm trying to make, and I really want to make sure I understand what that's going to mean for what I'm going to need to do downstream. Now that we're back under the legacy regulation for USDA, we're no longer counting edits. We're no longer talking about, is it an indel? Is it a base pair substitution? Did I use base editing? How many DNA strand breaks did I have? So I think for USDA, we're in a good place there. But then the next question is, well, what does your product look like to FDA? What does your product look like to EPA? And maybe you're not going so far that you need to go through those agencies as well. But they do not have that standard definition, at least across the 3 agencies in the US that regulate biotech.
I want to follow up. So there's a requirement in the US that anytime you sell food, it has to be safe and legal. So do what plant breeders have done for the past forever. Do you have a risk hypothesis that would indicate that modification would create some sort of a hazard that needs to be assessed for risk? And if you don't have a risk hypothesis here, that's it.
Yeah, so let me point out real quick. So the FDA regulations, as Wayne said, your food has to be safe and legal and all that sort of thing. FDA regulations technically are voluntary. However, if your product gets in the food supply and it's found in the food supply, you better have a lot of data showing that it is at the very least equivalent to other food in the food supply and doesn't pose that risk. Now, FDA tends to look at a couple of things. They look to see that you've fully characterized what you've got and you're not interrupting any genes that may inadvertently produce some unknown new protein or something like that. And they're generally looking at the gross chemistries and constituents that one looks at for food safety. So your vitamins, your fats, your carbohydrates, and what's known in terms of potential toxins that the plant produces. Anti-nutrients, that sort of thing. So there's some basic information that's really should be fairly obvious what it is that FDA needs to know. But you do need to molecularly characterize what you've got also.
There's consensus compositional lists for most major crops.
I was just going to add too on the FDA piece. FDA recently put out guidance for their voluntary expectation for anyone looking to enter into the food supply, plants produced using genome editing for human consumption, and what they would hope to see from a developer, again, from a voluntary standpoint. So they have these voluntary pre-market meetings. There should be a list of them on their website, but due to reductions in staff at the agency, they're not being posted. If you are considering this pathway, I would highly encourage you to read through that guidance document because it's maybe not as intuitive as you would think from a scientific perspective. Going back to one of the questions earlier, FDA put a lot of caveats into some of the guidance they were giving. Again, voluntary guidance, but guidance nonetheless.
Okay, question.
So I'm Liza Lockwood. I am the medical affairs lead for the crop science division of Bayer. And I get very involved with the sort of communication of science. What do you think is the difference between how this has been communicated in Europe and the US versus the successes that we're seeing in Latin America, Colombia, Argentina? Is there something that they're doing that is making the case a little better than we have?
From the regulatory policy perspective, countries like Argentina, Brazil, they have different regulatory triggers and had an opportunity to place a checkpoint above their biotech regulatory process and say, is the product you've developed, the final product you're going to put into commerce, is it a living modified organism? Is it equivalent to conventional breeding? And if that answer is yes, you follow the conventional breeding pathway, whether it's a plant, an animal. The US system's not quite set up like that, so from a policy perspective, we're still struggling with the statutory authority given to our 3 agencies and how to fit biotech into that authority. On the public perception piece, and I'm not sure if that was your question too, I think, and I can't speak to what public perception looks like outside the US, but in the US, I know we've been really with these next generation products, whether it was genome edited mustard greens or even the transgenic pink pineapple, transgenic purple tomato, leading with what resonates with consumers, not talking about how amazing the science is, but what's the value to consumers. And for the pineapple and the tomato, I think getting them at a price point where people can buy them. The pink pineapple is no longer $50 and exclusively purchased online. I can buy it for $5.99 in Food Lion. It's now accessible to the public.
I'll give you an anecdotal response to that in that, so similar to what they're doing in Latin America, Japan has taken an approach where if you don't have foreign DNA or transgenic DNA, you're not going to be treated differently to conventional breeding. And they have 3 approved fish on the market there. And interestingly, they were developed by a researcher at the University of Kyoto who said, oh, these are cool, I'd like to commercialize these. And they went to the government and said, can I commercialize these? They said, oh yeah, no transgenic DNA, off you go. And it went to market, and there's been not the expected pushback that there was with GMOs when they were coming in from large multinational companies. And that gets to the fact that if you have a regulatory that is workable for small, medium-scale companies, then you open it up to things like academia coming in with little startup companies and editing a tiger fish, which is ironic because the puffer fish that they edited is actually really poisonous. Yeah. So you would think whilst they were in there, they would have just knocked out that toxin gene, right, in addition to making it grow fast. But anyway, but it's there's a really interesting paper there on why there's been acceptance in Japan of that when there wasn't previously. And one of it was altered regulatory approach, who's developing it, and then public attitudes becoming more interested in sustainability of food production and understanding that this is a tool that can help with that. So it was kind of a tripartite change of attitudes in Japan.
Are we trying to model anything like that?
What's that?
Are we trying to model anything like that to maybe see if we can change public perception?
It would sure be nice.
I mean, Norfolk Healthy Foods, I mean, I'm growing the tomatoes in my backyard. I know they're for sale in Food Lion. I saw the pink pineapple finally came to a reasonable price. Yeah, because it was last year, it was expensive.
Yeah. I've got a few tomatoes in my garden too. So.
Yeah.
But are we trying to model these sort of things? It would be great if one could do that, and the U.S. government has tried to do that. Speaking as a former regulator, there are laws in place that we're kind of stuck with. And unless Congress can get together and change the laws, and we see how well Congress generally works together in terms of agreeing with each other, it's very difficult to do. So you're stuck with the laws and the regulations that you've got. And they literally, when they originally put the biotech regulations in place, they really had to try to fit what they wanted to do into the laws at the time, and then they wrote the regulations to fit as best they could at the time into the laws. But they're still stuck with the same basic laws, and it's very, very difficult to get these laws changed. So.
Yes.
Hi. I think this question maybe is good for Wayne and John, and we are working on some rootstocks and also on indoor plant. And I wonder if there's any difference for both rootstock and also indoor plant for the regulation and for the transgenic plants, or maybe the gene editing plants.
Yeah, I mean, if you're dealing with indoor plants, so these are house plants. Is that house plants? Yeah, I mean, again, depending on what you've got, Currently, if you want to move it across state lines, you have to have a permit or a notification. If you're not releasing it to the environment, even with an indoor plant, presumably it's going to be tropical. We still have tropical locations in the U.S. We have Florida, we have Hawaii, we have Puerto Rico, we have Virgin Islands. So things presumably could be grown outside there. So I want us to consider that. If you're just doing things in labs, I think it's not so complicated. You don't need permits to move things. You don't need notifications to move things. Again, even the regulated process is not that bad. And that's under the old regulations. It's an interesting process. It didn't really get to the point where it caught on until people started kind of doing gene editing, genome editing. Then it became much more useful because people were it's much more easy to remove all these plant pest sequences, your sequences from Mycobacterium and what other various other sequences there might have been in plasmids that you're using to do transformation work with. So we can talk about it more if you want and try to. A lot of times there's nuances to the regulations, and I know there's nuance to the research that people do, and there may be things that researchers can do to avoid some of the regulations if they want to.
Yeah, and it goes back to what John was saying about using existing laws, like using, USDA, plant pest and definition of plant pest. So, if your DNA does not come from a plant pest, and if you don't use a plant pest, i.e., agro, to do your engineering, USDA regulations don't cover you.
Some of the first products that were kind of not regulated without having to go through the whole petition process were used with linear DNA. They didn't have any agrobacterium sequences, and they used biolistics to get them in. And so those were some of the first things that USDA looked at and said, yeah, we don't need to regulate this. And it was a relatively short process of literally just asking the agency, not even having to submit much information to them. Say, biolistics, no agrobacterium, you're good.
Yeah, so Freddie Alpeter, University of Florida. So if you work on some of these non-regulated through the MI-regulated process, and you want to do a field trial, and there is some triggers in it, so they don't have plant pest sequences, but they have a trigger for EPA, but you're staying under the 10 acres, and then after you're done with your field trial, you walk away, but these persist in the environment since you didn't have a permit, right? You didn't pay attention to that. So now something persists in the environment that has a trigger for EPA. It's not 10 acres yet, but maybe 10 acres after a few years, right? So what is your liability in this?
I'm not a lawyer.
I'm not a lawyer.
But it's a good point, though. I mean, it's a good point. Again, even if you're doing things that may not trigger the regulations, you maybe should have some stewardship processes in place to make sure you get these things under control and seed clean, as Ray said.
I would highly recommend that. And then the other question I had was, do we have some sort of an idea if this MI-regulated process is going to stay for the long term, or are they actively working on replacing this with a secure 2.0 rule?
So the MI-regulated process is a discretionary process that USDA has put out as a way to better engage more transparently with developers who are asking this question and then put out responses that the public can see. The MI-regulated process is not hard-coded into the regulation, so there'd be no reason for USDA to try to rewrite it today. or to take it away, because it's really not part of the regulation. And you can always go to your government agencies and ask them, do they have oversight of your product? This was just a nice way, the Am I Regulated, to be transparent. So to your first question about what if there's some EPA oversight under FIFRA for something I want to put into the field, you could go and talk to EPA about that. You might even want to talk to the state you're in, because one of the nice things when there was oversight by BRS and EPA for field releases is they would coordinate any state-specific requirements too. So that's just something else you might want to think about. But you might have some more thoughts on Am I Regulated?
Yeah, I would like just to chip in as somebody who's been on the receiving end of having to clean these things up. Not only the public ones, but some others as well. Really, if there's any chance that this could be regulated anywhere in the world and you put it in the field, please do a complete stewardship program. Because even if you're a university and this gets to Europe and it damages trade, there's somebody out there that will be interested in coming to see you and maybe want to, sue you. So please be very careful when you're putting stuff in the field, not only if it's regulated in the US, but if it's regulated anywhere. Do a complete stewardship program. Make sure that your people are trained, make sure that you know where the material's going, and you follow it. Please. That would be my advice. Yeah?
Yeah, one more thing.
Just to follow up on my second question, so we don't have any indication that they are coming up with Secure 2.0 anytime soon, right? Or are they?
I don't think we have any indication, given what Nicole said about USDA deciding not to challenge not to appeal the decision. My guess is that they are doing some revisions to the regulations as they were written. There were two things. There were two things that the court ruled on. One of them was the Administrative Procedures Act. That is literally a paperwork exercise. And the other one is decisions under, I think it was the Noxious Weed Act. There were some, and the judge ruled against the agency in both cases because they didn't talk about things enough. These were things that the agency thought were kind of settled issues, so they didn't talk about them in their environmental impact statement. And I mean, one could could at least in theory cure the judge's issues under the Administrative Procedures Act and the Noxious Weed Act fairly easily. However, there are lots of other government regulations that the judge did not rule on, one of which is the National Environmental Policy Act. And usually government agencies who make these large decisions about new regulations, about huge projects, have to deal with this National Environmental Policy Act. And it's not uncommon for government agencies to lose decisions under the National Environmental Policy Act. But the judge never even got to those in this case. So, they may be able to cure the two issues that the judge got to, but the judge still hasn't looked at the National Environmental Policy Act issues. So
I want to come back. There was questions earlier from other countries, etc. And we had a question here about in traceability. If we were required to do traceability instead of detection methods for gene-edited products because they're difficult to detect in commodities, what would the effect of that be on commercialization of these products?
It's a hard one. When we look at the regulatory policy perspective for how genome edited, let's say, plants are going through the USDA process or processes in Argentina and Brazil, the determination at the end of the day is that this genome edited plant is no different than something that could have been developed through conventional breeding. And I think then as the developer community to ask yourself, well, what would we do for traceability for a conventionally bred product of this type? And to start there, we know that if you're breeding onion seeds, you have traceability in your program. You can do that varietal differentiation for your production, for your certification. Is that sufficient then to also encompass your genome edited material? And then what does that mean as you then trade into countries that have a slightly different regulatory process? So I think from our perspective as the industry, these are things that get captured as part of quality management systems, stewardship systems that companies develop, and not necessarily something that the government has to have a hard set policy on today.
Yeah, to me, it's much easier to monitor for a transgene where you can use an antibody test strip and sample along the entire chain of from harvest to consumer than it is to sample for a change made to the DNA that may not be coding for anything. You actually have to do DNA extractions and PCRs and everything else, so your high throughput is going to be a lot less. Your costs are going to be a lot higher, and at the end of the day, you still don't know if it's a natural mutation or an edit. So if you want traceability with edited products, it's going to be logistically, I'm going to say, impossible.
Animals?
Well, I guess
traceable, a lot bigger, right?
Yeah, well, and also because milk and meat, it's a combination of thousands of animals. That's all going to be quite complicated. And I guess I just look back to the history of recombinant bovine somatotrophin, which for those of you that weren't around, that was an early genetically engineered pharmaceutical given to dairy cattle to increase their milk production. And there was a lot of, Jeremy Rifkin at the time created a lot of fear around that. And so certain creameries said, okay, we're going to label our stuff as GM BST free. And if you want to supply to our creamery, you may not use that technology. And effectively that spread, and it's taken over the country now, and that product is off the market. That's, I think, what will happen, because you won't be able to comply with being able to guarantee it's not genome edited, and so you'll kind of have this de facto precautionary position of, well, let's just not allow it in the food supply in case we get consumer pushback. And I wouldn't be surprised if we see that even now with the genome edited pigs that are disease resistant, that there's going to be some concern from producers that let's not adopt that pig, let's keep having the sick pigs because we don't want consumer pushback on pork. Yeah. So I think if there's a requirement that it has to be traced, that basically creates two entire supply chains, and that's going to be cost prohibitive, I think, and it'll really disincentivize the use of the technology.
Going back to seeds real quick, there is what's called the Federal Seed Act in the U.S. It falls under USDA, and it defines various qualities, and this is mostly commodity seeds, so your corn, your soybeans, cotton, that sort of thing, and it defines the percentage of off types that you're allowed to have in a seed lot in order to reach a certain type of certification that is described in the Federal Seed Act. The thing is about these off types is that most of the assessment for the off types is a phenotypic assessment. It's not based on DNA. It's not based on most things that you can literally see in the field. It is based on things you can see in the field. It's not based on any sort of chemical analysis of the plant itself. So, to try to deal with genome edited products and looking at phenotypes in the field is, as Wayne said, nearly impossible.
There was a question.
Yeah, more of a comment, I guess. I just want to clarify. We talked about all these exemptions and things like that, and we say if there's no foreign DNA. Your Cas9 is foreign DNA. If you're working on a perennial crop, if you haven't segregated out your Cas9, you put it in the field, that's an environmental release, that's illegal. You need a permit to do that. So people working with perennial crops need permits all the time, like if you're putting a tree crop out there or something like that, if they haven't segregated their Cas9. Now, there are methods to do that without Cas9, RNPs, better ways to segregate out and things like that. But pay attention to that. Just because there's a rule, you don't apply because you haven't segregated out your DNA. You do have foreign DNA in there. Cas9 is not plant DNA. Things like that. So that one of the comments. I did have a question about. I'm a little bullish about consumer acceptance for some of the things it said, and I cautiously say that. I start working on this in 1993, and I said in 1995 it'll all be great. Well, here we are. But the Impossible Burger is a perfect example. That's transgenic soybean. It sells just as well as the Beyond Burger. So that's a good product. People don't care. Give me a good burger. You know, I have that hope for the purple tomato. But I want your comment. Do you think, you know, the news we had this week of the baby coming out of a Philadelphia hospital, you know, with the CPS1, you know, that was gene edited and, you know, really fast response, looks, you know, things are looking really, really great on the human side, will change things.
We tried that with insulin, right? Genetically engineered insulin, that's what all diabetics are pretty much using. So medicine is different to food, I think. I hope your optimism is justified, Alan, and I would love to be celebrating that too. But I think it'll depend on what competing market forces do, and the fact that the non-GMO project has already said that they're going to label for gene editing and create fear around this technology makes me think that that's a very successful business model they have running, the butterfly label. So I would not be surprised if they don't try the same thing here.
Yeah, I'm likewise cynical. If you remember back when, say, the Flavor Savor tomato came out,
Yeah.
it was not a very good product, but it sold really well. People were willing to accept it. But then, you know, Starlink happened, and public opinion totally switched overnight. And it does not help that there was a buck to be made by groups that were telling people that this is dangerous. And I mean, we're at an all-time high of groups saying that everything is dangerous, and people are not nearly as trustworthy of scientists now as they were 20, 30 years ago. So I think that's why I said earlier, I think public acceptance is a very fragile thing.
Going back to that CRISPR-Cas9 comment, you're right. And I actually submitted a notification to BRS for a product that we described as gene edited. What we didn't say in the notification was that the CRISPR-Cas9 DNA had been segregated out. And they kick that back to me and say, "Oh, you didn't say that this has been kicked out." So we're assuming it's still in there. You haven't bred it out, so we're assuming it's still in there. So you have to go under a permit, much more rigorous under a permit for APHIS BRS regulations. If you're doing the CRISPR-Cas9 and you're able to breed the material, segregate it out before you want to do a field trial or something like that, do it and say it very clearly in your application. It will qualify under notification if you can say that. If you can't say that, then you're going to need a permit from APHIS BRS. And
can I just a quick comment on that? You want it out because it's still working.
yeah.
You'll get new edits, new generation. Every meiosis is a new generation, new edits. So it's not going to segregate 1 to 1. It's going to come in new edits as well.
Yeah. Okay. You got a question?
Okay, my name is Kavita. I'm from University of Minnesota. Following up on this, so as a researcher, how can we effectively work to change these negative narratives? And there is a lot of misinformation about GMOs and gene-edited products, especially in like the food we consume. So how can we communicate to change these negative narratives? I want to hear your thoughts on this.
So we recognize that importance, and we recognize that consumers get to vote with their dollar, and consumers don't vote based on how cool the science is and how great we think these products could be. Talking to consumers on their level, understanding what is it that motivates them to buy an organic product or a non-GMO project certified product, why? What is it that could help them make a different decision? I think too, we talked earlier about the purple tomato and the pink pineapple and the genome edited mustard greens that we've seen in the grocery store. Those are still really a novelty, and getting people excited about products like that, so when the time comes that we need these tools to save bananas, to combat citrus greening, to help with a wheat blight disease, then there's less of that concern. Then people are more willing to accept, oh, well, we liked the pink pineapple, maybe we'll like these genome edited oranges as well. I think too what we've seen in the very little bit of research we had just started to do at Bio is that younger generations tend to be a little bit more receptive to the conversation, to the use of technology. You bring it back to some sustainability objectives. People seem to really resonate that, and people really appreciate the conversation starting early. And so that's some of the things we've seen.
Yeah, seeing is believing. Once the flavor saver tomato was gone, and all the rest of the crops were unseen things that were out there somewhere but could come back and find their way into my food. And as we have seen with, when people were willing to pay $50 a pineapple and sell out, or the purple tomato at $2 a seed, or even the glow-in-the-dark petunia that's selling for $30 a plant. There's a novelty aspect, but more important is things that people can touch, feel, and taste. The Arctic apple, the Simplots, the white russet potato, that people can actually feel. If you can feel it, taste it, touch it, you can decide, yeah, it's safe, it's good. But, you don't know much about the soybean lecithin that's in my cereal. It's much more abstract for most people. So make it real. Let them be able to see and touch it. Orange petunias, the moon dust carnations. I think those are the type of products that will build trust.
And one more question here. So come up against time.
Hi, my name is Marina Corona. I'm entering my last year as a PhD candidate at UC Davis. That was actually my question as well. I do a lot of outreach, and I think a lot about science communication. So I appreciated your answers about how to approach it and relate it to something you can feel, touch, taste, to make it a little bit more accessible. I guess because my question was answered, my experience with it has been science is, of course, we all think it's really amazing, but I notice sometimes for people who maybe find the science less relatable, try to meet them where they're at. If they're scared for health reasons, discuss with them why they're scared, and then bring back things that maybe they do know about, like insulin, that has made such a positive impact on our society. And maybe I found that there can maybe be a little bit of success with at least opening minds that way. So more of a comment than a question now because it was answered, but just wanted to share that.
Yeah, science communication is tough. There's no doubt about it. I deal with it in my own family, and it's hard to do the things and there's papers there's social science papers out there on this, so you can read how to have these conversations. You need to bring out the facts, be respectful how you have the conversation, basic things like that, but always stick to the facts too. So.
Yeah, if you're going to be communicating science, you need to have a great slide deck with wonderful, colorful, friendly pictures. Most people are far removed from agriculture. They don't really know, showing a GM cotton next to non-GM cotton or a GM corn. Those things really carry a lot of weight. I always start with the most familiar things, usually the flowers, some of the things they'll find in their kitchen. I like to point out that who has washed their clothes in cold water recently, and that's because we have, engineered enzymes in there, and point out how, much the process is used in wine and cheese around the world. Who likes light beer? Well, guess what? And candy cordials and vitamins, and say, we don't really worry about these. It's just we worry, about the food, and it sort of opens the stage for people to move on to other parts.
One last question.
Yes, thank you. So I want to touch the 2 points, and they are mixed with questions and comments. One is about the global public acceptance, because one of my activities that I'm connecting with students from all over the world, and sometimes topics comes to, discussion comes to this topic, and the main thing I hear from people is that, so they say, okay, like we didn't know about this, but it sounds amazing. It sounds like a dream that everything is so controllable, so precise, and so fast compared to conventional methods. And then they are questioning why there are so many questions and problems in the regulations in this. So that makes them question it, even if it sounds so good. But if it's not accepted by still so many countries in the world, by the majority of the countries, then they question it, then there should be something wrong with this. That's where the mind goes. And my other comment and question is, this is more like a question is, so for instance, I know some companies that in my home country, I met them, and there are some gaps in the biolaws that it talks about on genome insertion. So there are a few companies, they are already making commercial their products by using this new genomic techniques, and they are not making this like everyone knows about this, but they don't have the label, they don't have to go through like different regulations because they don't apply as how they breed these plants. So I was wondering, where do you see it's going in maybe next 10 or 15 years? If there are more companies doing this kind of activities and taking advantage of these gaps in the regulations. And yeah, thanks so much.
So I think one comment was, do more complex regulations make people trust it less? Because my God, it must be really dangerous if you need that level of regulation. And I think every one of us would agree 100%, and the regulations are ridiculous at the moment. However, they are what we're dealing with. And we were told, oh, if we get the FDA approval for the fish, that'll give consumers confidence. And it took 30 years, and it didn't give consumers confidence because it took 30 years to get through regulatory. So to your point, I think that's absolutely true. I didn't quite catch the second question. That was doing gene editing without?
Yeah, without any like information. I mean, they're doing it. It's like illegal, but it's not illegal too. Like, do you see my point? So there's a gap in the constitution, so they just take advantage of this technology to develop.
If you did gene editing and didn't tell anyone, do you still need to get regulated? Is that what you're asking?
Yes, they are using it, but they are not applying as they are doing it that way, and they're not regulated because it's not detectable.
Well, that's a risky question. I think when you do have regulations that don't make sense, you'll tend to have people that respond accordingly. And the one I bring up there is clones, clones which are exact genetic copies, are banned in some countries, and it's like literally, how could you tell?
Yes.
And yeah, I'm going to sell it. Oh no, that's not a clone, because you can't tell. And so I feel like if the regulations are not sensible, that you will get situations like that. And at the moment, I don't think they're very sensible, at least in some countries.
I had an interesting discussion a couple of years ago with the head of the EU laboratory that tests for genome-edited products. And the discussion was something like this: well, I've got 2 ducks, and one of them's edited, and the other one's just bred, and they're exactly the same, and I have to regulate one of them, not the other one. How can I tell the difference? I said, you can't, but he says, I have to. So think about the ridiculousness of that situation that we're getting in. So we've got one that's bred exactly the same mutation as what's in the population, but this one has to be regulated and that one doesn't, but you have no way of telling the difference. And that's one of the problems we have with doing regulations based on process instead of the product. Yeah.
For those who haven't been around a long time, the regulators have been talking about regulating product instead of process for about 40 years.
Yeah.
And it still hasn't come to fruition. So,
yeah. So given that, can each of you give me an example, if you can, of a successful, or if you have to, unsuccessful, regulatory process regarding gene editing organisms? So some example that you want to talk about.
I think Latin America, with if there's no foreign DNA and you could have achieved it using conventional breeding, you're conventional. Goodbye.
Thank you.
A successful example I have in the microbial space for a genome edited product is a nutrient enhancement. It's a genome edited microbe that helps with nitrogen fixation for plant uptake. It can be applied as a seed coat, or it can be applied in furrow in the field. And you go from having to put a synthetic product that has a lot of weight behind its liquid formulation into just a granular or a seed coat. And I think that product is really fantastic because it did get to go through the regulatory process pre-secure rule, so pre-2020. Otherwise, it would have gotten caught up in some of the things we've been talking about today over the last 5 years. And this product has now gotten to be in the field, be used by growers, have some data behind it to show it's effective, to show the effect it has on the soil health. And now, hopefully, that we're back to that legacy regulation, we'll see more similar type microbial products being used, because I do think that's a really great success story. And again, if we were under the secure rule, that wouldn't have happened. So I do think we have now that potential again.
I'm going to vote for the secure rule that got thrown out and the way it handled transgenics. I think that really lowered the barriers to something much more reasonable and can measure it with any issues that might have been present. And it just was much more science-based, and it really streamlined and served the process, enabled things to get through it.
I haven't got anything right now. Go ahead, Ray.
Mm-hmm. Okay. Well, given that we're out of time, let me first thank the panelists for the discussion. Thank you, everybody who asked questions and comments. And thank you again to the Bragg system for funding this, and for SIVB putting this on. Did I forget anybody? Can you run that last slide I gave you about the session tomorrow? Tomorrow there's a session again on biotechnology approaches to risk assessment, and be there, be square. It's 8 o'clock in the morning. Anything I forgot to do? No, thank you very much.